That's a major slide, and I'll spend majority of my time on this slide. Let's explain workflow procedure how to do relative quantification of literally dozens, maybe hundreds of proteins between two samples. So we call it sample A and sample B. It does not matter what you want to compare, like cells control and cells treated, or disease conditions versus control, or disease plus treatment versus disease no treatment. We're just talking about relative quantification between two samples. And all what we do in vitro, we modify proteins in one sample with light compound, and in other second sample with heavy compound. And in this case, we use acrylamide. It's a very simple compound. People used to use it to run for the acrylamide gels, but it has double bond. And you see, commercially available heavy acrylamide has 3 deuteriums shown in blue versus 3 protons. The only difference is basically that it's 3 units heavier. That's all what we need from mass spec. We modify cysteine residues because of alkylations with a double bond. In sample A with light compound, in sample B with heavy compound, and mix it at 1:1 ratio. It's something like based on total protein, like 10 milligrams of total protein from one sample and 10 milligrams from another. Then we come into a point that we have to, at some moment, generate peptides, and to separate either proteins on the level and then generate peptides, or best to generate peptides and separate peptides. It doesn't matter. It's quite a lot of options how to do. I show you just like one workflow. In this case, it was 2D gel. And every spot, what you see on this 2D gel, is basically a mixture of this specific protein, which came at some proportion from sample B and sample A. They just co-migrate. And then we took a spot out. We do in gel digestion and generate peptide. In this case, it was used multi-TOF mass spectrometer, again, could be something else, to identify protein. Based on these peaks, we identify and we got a name. And then we go just like zooming spectrum and searching for cysteine-containing peptides. And what happened, every of these peptides would be represented by 2 shifted isotopic envelopes and shifted 3 units. Because difference between these compounds, 3 units molecular mass. What happened, basically, you see here. This isotopic envelope represents this cysteine-containing peptide from this protein from sample A. And this isotopic envelope, shifted 3 units, represents this peptide, which came from this protein from sample B. And we just got a ratio of area under the peak, and we see that ratio 2.5, but we mix it 1:1. And we draw a very simple conclusion that this specific protein was 2.5-fold up regulated in sample B versus sample A. Of course, I may take all of these spots in one experiment, and in one experiment really assess relative quantification of several dozens of proteins. So what happened, as I mentioned in the very beginning of this talk, whole idea was to compare premature senescent cells versus control in terms of, again, which proteins were up, down regulated, or not changed. But on the level of different steps of separation of these proteins after modification, we notice that some fractions are really enriched with fragments of these proteins, of some proteins. And it's rather large fragments. It's not peptide level, yes, it's you see, for example, this is blue gaptage, 36,000, and we have dozens of fragments which is lower in molecular mass, but it's still big proteins and they might be properly folded. That's a quick example just to prove that yes, we do quantification for specific proteins. That's the name of this protein. Quantification was done based on 2 different peptides, just like to show that it's more valid. We got obviously for full-size protein some decrease in this ratio, and obviously increase for some fragment. It's again just comparison of area under the peak between these peaks. So for GAPDH, that's, you see, showing in red, it's a full-size protein with almost. With one unit, ratio 1, which now change, and the tons of fragments, which obviously some 2-3-fold up regulated. Not up regulated, increased. So basically, that's the last slide in my presentation. And I have to mention point number 1. That so far in literature, it was published 3 well-known, sorry for misspelling, lipofuscin irreversibly damage mitochondria and aberrant proteins. It's type of waste material which people used to see accumulated in post-mitotic long-living cells. What we basically reporting here is that we see, I would call it to number 4, accumulation of large protein fragments of typical cytosolic proteins. So, and point number 2 probably is the most supposed to attract attention, because apart from ideas that it might be just a waste material, yes, these fragments could have a specific activity other than full-size proteins, because it's still very large. It might be properly folded. There are stories about translocation of proteins like this in nucleus, where they may play a role of transcriptional factor. Enormous localization to membranes and so on. I guess I'm done. Thank you very much for your attention.